back, or we'll just start in a couple of minutes. Okay. Hi, everyone. Welcome to the afternoon session. So this is session one. This is the current advances in laboratory-based, lifestyle-related cancer research. My name is Gerado Mackenzie. I'm an associate professor in the Department of Nutrition at UC Davis, and I'm going to be presenting one of the talks alongside with two fantastic graduate students that will come after my first presentation, and I will introduce them then later. See if this, do I press forward? Okay, so today I'm going to discuss a little bit about some research, some recent data that we have been working on the liver safety profile of a ketogenic diet in combination with gemcitabine, a chemotherapeutic used in pancreatic cancer, in pancreatic-bearing mice. And this work has been done. Oh, sorry, I have no conflicts of interest, And this work has been, again, led by a graduate student in my lab, and now she's a postdoc in Italy, Natalia Cortez. And just to give you a short introduction, pancreatic cancer, for those that are not very familiar, is a lethal disease. Yes, as you can see here, this is maybe a little bit older now, but still, among many other solid tumors, pancreatic cancer still has the lowest 5-year relative survival percentage. And similar to what happens with liver cancer that we heard earlier today, the projected cancer deaths related to pancreatic cancer will likely become the second leading cause of cancer deaths by 2030, just lower from lung cancer. So clearly, new therapeutic strategies are needed for this cancer. And to give a little more perspective of why pancreatic cancer is so difficult to treat or to diagnose, again, this is a partial list of many of the problems that we face in pancreatic cancer. So only a small number of patients, I knew I was going to mess up with only a small number of patients present with resectable disease, and currently surgery is the only kind of cure, or somewhat cure for pancreatic cancer. So given the location of pancreas and the problem with some of the arteries that go by, and the problem that metastasize so early, like many patients, again, are ineligible for the surgery. So another problem is, again, diagnostic to detect and follow patients is lacking. There needs to be a little more work on this. Patients' health decline very rapidly, and therefore they're ineligible for clinical trials. And we all know, of course, if there's no clinical trials, it's very difficult to advance science. Then PDAC incidence is increasing. We heard yesterday about the incidence of cancer and why that's happening. Maybe, of course, we are preventing other diseases, and then, of course, as cancer is a disease of, many cancers are a disease of aging population, the incidence could be increasing because of that. Other suggestions could be the aging population, the rates of obesity that are increasing, or maybe changing in the microflora due to diet or other aspects. Another point that I want to bring up, pancreatic cancer maybe has a unique microenvironment compared to other types of cancers. And this is particular for pancreatic ductal adenocarcinoma. Yes, and here I have an image of a trichrome staining, and where you can see the cancer cells they're lower in number compared to the huge and dense microenvironment. And there's several speculations that this maybe it's limiting the penetrance of drugs towards it. It produces a hypoxic environment, and maybe some drugs to treat pancreatic cancer cannot reach the actual tumor to treat. Yes? Again, this has been led to some clinical trials, and some of them, like targeting hyaluronic acid, again, like even though the concept was interesting, again, the patients died earlier, so they had to stop that. So sometimes maybe this is a response of the actual organ, of the actual body, to try to retain the tumor there and not try to lead it to spread. But still, this is still, again, very unclear. So those are some speculations. Other problems that we face in pancreatic cancer is that it's an immunologically cold type of cancer. So immunotherapy has not been effective in the case of pancreatic cancer. Only 1 to 2% benefit from immunotherapy. However, of course, this is a very active area of research. And the other problem of pancreatic cancer is it has the highest incidence of cachexia. And it's usually around 80% at time of diagnosis. And this is another aspect of what we were interested in because of some previous work in our graduate group, where they've shown that pancreatic ketogenic diets could be beneficial against cachexia in aging mice. So we thought, okay, maybe the ketogenic diet, by improving the quality of life, could potentially be a beneficial way to add to adjuvant therapy for pancreatic cancer. So again, why a ketogenic diet? I mentioned the last step, but this is again some of the preclinical and clinical applications, and this is a very active area of research. You've heard from Dr. Candley an hour ago about the potential use of ketogenic diets for maybe lowering insulin levels as well. So there has been successful use on epilepsy in children, so it could be easy translatable into humans. There's a lot of preclinical studies showing that it's effective. There's some evidence of clinical evidence that ketogenic diets are also effective in humans. So again, and the research showing that ketogenic diets and ketone supplements could be beneficial for increased muscle strength. So and regarding this point, many of the studies have been used ketogenic diets in animal models, have been usually used xenograft models. And xenograft models either orthotopically implanted or subcutaneously implanted, and those do not reflect the actual disease and this unique microenvironment, for example, that is occurring in pancreatic cancer. So they have some benefits, but they also have very important limitations on their use. So these are some of the preliminary data, or published data, I would say, on the role of ketogenic diets in pancreatic cancer. So here you can see that this was a study in 2014, orthotopic models, and where they've shown that a ketogenic diet was beneficial, reducing tumor growth and tumor volume, in orthotopic implanted pancreatic cancer tumors. In this one, ketogenic diet alone showed no benefit in increasing survival in the Mia-PACA tumor cell line. However, when combined with radiotherapy, with radiation therapy, they showed a beneficial effect. This one is the one that Dr. Candley presented, that they were published in 2018, where they can show that ketogenic diet in brown, there's a beneficial effect when combined with a PI3 kinase inhibitor. And however, ketogenic diet alone show, again, minimum benefit. Yes, and interesting, again, as we got closer to our study, that of course it was delayed due to COVID, but many studies were delayed. This publication came up in medicine, in medicine published, showing that a ketogenic diet synergizes with cytotoxic chemotherapy with with paclitaxel or Abraxane, gemcitabine, and cisplatin. They've shown in subcutaneous xenografts a beneficial effect of the diet plus the chemotherapy, the triple chemotherapy. And also in a small cohort of KPC, on only male KPC, they also shown a significant benefit of this diet plus the chemotherapy. So based on this, again, we started this with my graduate student like some time ago now. Our interest was to evaluate the effect of a ketogenic diet alongside with chemotherapy in the KPC animal model. Yes, and for this purpose, we use this KPC animal model, this genetically modified model that carries two mutations that are mostly commonly expressed in pancreatic cancer, the KRAS and the P53. And this is the clinical relevance of the KPC as a model for pancreatic cancer research. I just mentioned two of the most common mutations that are observed in pancreatic cancer patients. The sites of metastasis are similar to what is observed in humans to the liver and lungs. It's an immunocompetent mice with a possibility of evaluating maybe immunotherapy in these animals. The histopathological resembles, like part of the microenvironment, for example, shows very similar aspects to the humans. The mice health declined very rapidly. Usually, like, these animals die approximately at 6 months of age. And these mice develop cachexia as they age. So we could potentially use these mice as a way to evaluate anti-cachexic treatments. So this was our first experimental design. So we had mice. Once we confirmed the genotype of the mice, around 3-4 months, we detected the tumors by ultrasound, and then we randomized into the different groups. So we had the control diet, either a ketogenic diet, a control diet plus gemcitabine. This is going to be CG, is the control diet plus gemcitabine treatment. And the KG is the ketogenic diet plus gemcitabine. And these will be the colors that we will try to maintain throughout the presentation. So blue is going to be the control diet bars, ketogenic diet, the red, and so on. And then throughout the treatments, we performed several studies. We collected fecal sample at baseline at 1 month and every month. Then we measured beta-hydroxybutyrate levels as a measurement of the ketogenic diet, or ketone bodies, ketosis. And then we also determined grip strength as a muscle strength, as well as body composition for these mice. And then when they reached survival, we sacrificed the animals and collected the tumors. So, of course, feeding a ketogenic diet, as you can see, enhances the beta-hydroxybutyrate levels after 1 month, and actually this occurs very rapidly. 1 week, 2 weeks of diet, this is a strict ketogenic diet, they already increase either alone or in combination with gemcitabine, and this was observed in both female mice and male mice. We've enrolled around 16 animals per group in these studies. We determined the glucose levels. We've observed a reduction when we combined. These are all the animals combined. However, this was basically observed in male mice. And this, again, but it was not maintained throughout the treatment. So regarding body composition, what we observe is that this is the body weight on the top, and this is the body composition in the bottom, the lean mass. And we observe a reduction in the lean mass for the gemcitabine-treated mice, the purple and the green bars. And we think that this is related maybe to a little bit of response to the chemotherapy. The chemotherapy was given at 100 milligrams per kilo. That's something that is given in previous papers. But these animals, again, maybe there's some toxicity at some point for these. And then, however, these animals recovered after they finished the chemotherapy schedule. Yes, these were just given 7 injections for twice per week for 3 weeks and a half. So again, I think the most interesting data is the enhancement of survival that we observed. As you can see, ketogenic diet alone did not increase survival. However, in combination with chemotherapy, with gemcitabine, we observe a significant increase in overall median survival. And this, when we separate males and females, we observe that this reached significance, and I'm just showing the control diet and the ketogenic diet plus gemcitabine, just for simplicity. and this is significant in the females. However, it did not reach significance in the males. However, again, after consultation with a statistician, she said that this is not a sex-dependent effect. However, maybe it's the power, but this is still more evident in female mice compared to male mice. So how does the ketogenic diet could improve pancreatic cancer survival? We think that this could be different ways. Yeah, it could be throughout the muscle, and we observe an increase in muscle strength in these mice. Yeah, I'm not presenting this data today, and then also maybe a modulation of the microbiome or some direct effects on cell metabolism at the pancreas. So to try to answer these questions, what we performed is a mechanistic study in where we had just two groups, the control diet plus gemcitabine, as well as the ketogenic diet plus gemcitabine baseline, and then we sacrificed the animals at 2 months. Yeah, so we had the same time for all the animals, and we performed an RNA-seq data from pancreatic cancer tumors that we isolated, and like the gene set enrichment analysis show that these are some of the pathways, but some pathways that we were interested in were like fatty acid metabolism, as well as the PI3 kinase mTOR pathways, and as well as AKT. So these are other pathways we're interested in, like glycolysis as well, change, and these are something that we are continually investigating right now. So for example, ketogenic diet plus gemcitabine, and as a control we use control plus gemcitabine in these studies. So just to pinpoint what could be the effect of a ketogenic diet. And as you can see from this gene set enrichment analysis, this was significant changes, and even though we didn't observe changes in the whole panels of fatty acids, measured by GC, what we observed is that there was a significant difference in select specific fatty acids, and this is in the pancreas of the mice. Yes, and these have been shown, some of these, yes, palmoleic acid, yes, or palmitic acid, these have been shown to be related to some correlations with pancreatic cancer incidence. So this, again, still this needs to be confirmed. Yeah, this is just a measurement. We didn't follow up with these studies, and if these are key mechanistic studies. Yeah. Another pathway that we were interested in is the PI3 kinase AKT mTOR, and what we observed is that there was a reduction in phospho-AKT signaling, a reduction in phospho-ERK signaling, yes, a reduction in IGFR signaling. As you can see here, and this specifically, some of these were only observed in the female mice and not in the male mice. Yeah, so I don't know if maybe this is like a trend towards like why females are being more responsive to this effect or not, but this still needs to be confirmed. So we also evaluated the levels of the gut microbiome. Yes, for this we isolated in all the groups fecal samples at baseline and after 1 month in the treatment of the control ketogenic diet or control plus gemcitabine and ketogenic diet plus gemcitabine. And what we observed is that in baseline all the mice were still in chow diet. Okay, so there's no significant difference in many, even though here there was, but it is what it is. Yeah, what we observe is that there is an increase in the firmicutes to bacteroidetes ratio in the ketogenic diet plus gemcitabine group compared to the other groups, and a reduction in the alpha diversity after a month in the treatment of the diet plus the chemotherapy. Interestingly, when we evaluated like the linear discriminant analysis, and we compared, in this case, ketogenic diet alone or ketogenic diet plus gemcitabine, what we found out, and it's, I don't know, interesting that, again, as we heard like in the previous talk by Fessler, that feculobaculum was increased in the ketogenic diet plus gemcitabine compared to the ketogenic diet. And when we compare just gemcitabine in the control diet versus gemcitabine with the ketogenic diet, we observe also that this bacteria was increased. Yes, and we also evaluated that there was a reduction in the lactobacillus. And I find very interesting the feculobaculum because there has been showing in both colon cancer as well as breast cancer where that they've shown that this increase could have a tumor inhibitory role. And this, again, still needs to be confirmed, but I thought that it was, an interesting finding that after 1 month in the treatment, we've shown these differences. So as a conclusion of this efficacy study, what we were just published, we observed that a combined effect of a ketogenic diet with gemcitabine extends overall survival in the KBC mice. A ketogenic diet plus gemcitabine has a multifactorial effect, and this is, I think, a partial effect, inhibiting the IGFR, AKT, and ERK pathway, affecting glucose metabolism as well, and lipid metabolism in the tumors. And we've also shown that ketogenic diet alone and in combination with gemcitabine can modulate the gut microbiome that could potentially also play a role in the inhibitory effect. So, again, what about safety? Yes, I wanted just to present this data we just published, just to introduce yourself to, okay, we know that ketogenic diets are safe. However, in combination with chemotherapy, still this is unclear. Yes, and there has not been many studies evaluating the role of, or the safety of the ketogenic diet in combination studies. So from samples after the 2 months, what we've done is we measure the liver enzymes, many of the liver enzymes that are secreted into the serum. We measure the AST, ALT, ALP, bilirubin. Yes, we also measure some kidney parameters like creatinine, blood urea nitrogen. As you can see, when you compare the control plus gemcitabine versus the ketogenic diet plus gemcitabine, among all of these parameters, we didn't observe any significant difference in any of these levels of safety levels, both in the males or in the females. Given that a ketogenic diet is a high-fat diet, we then evaluated whether there was any fat accumulation in the liver and if this could lead to steatosis, or liver fibrosis. So as you can see from the H&E stains as well as the trichrome stains, we didn't observe any, and this was quantified by our pathologist in the cancer center, Dr. Matsukuma, and any of the parameters of liver fibrosis like fibronectin or collagen were not changed, or there was not any significant difference between the control plus gemcitabine versus the ketogenic diet plus gemcitabine. We also measured whether a ketogenic diet could affect any enzymes involved in fatty acid synthesis, and as you can see from the graphs, the only main difference or significant difference that we observed was in male mice, where there's a reduction in the levels of the fatty acid synthase. However, there were no difference in PPAR alpha or the strep BP1 or any phospho-AKT, for example. These are all in liver homogenates. We didn't see any significant difference in these markers. We also measured enzymes related to ketogenesis or glucose metabolism in the liver, and again, we didn't see any significant changes among the control and the ketogenic diet treated groups, both in the ketogenesis markers or ketogenesis synthesis, as well as the hexokinase or phosphofructokinase or PDH levels between the two groups. Finally, we also evaluated the role of a ketogenic diet in combination with gemcitabine with markers of inflammation and markers of oxidative stress. For that purpose, we measured TLR2, TLR4, and markers in the NF-κB signaling pathway, such as phospho-IκB alpha or phospho-P65, as well as the ERK pathway. And as you can compare here, the only difference that we've observed, significantly difference, is the levels of phosphorylated P65 in this whole homogenate sulfate. Related to oxidative stress, we determined the levels of 4-hydroxynonenal, as a lipid peroxidation marker, and as you can see, there was no significant difference between the control and ketogenic diet versus the control and gemcitabine versus the ketogenic diet plus gemcitabine treated groups. So, as a conclusion, the combined effect of a ketogenic diet with gemcitabine extends survival. Ketogenic diet plus gemcitabine has a multifactorial effect, inhibits IGFR, AKT, and ERK pathway, as we mentioned. Also glucose metabolism and lipid metabolism in the pancreatic tumors, however not in not in the liver. A ketogenic diet alone in combination with gemcitabine modulates the gut microbiome as we presented. And after 2 months of treatment at least, a ketogenic diet, it's safe, and a ketogenic diet in combination with gemcitabine showed no apparent liver toxicity. So what is next? Yeah, so that's why I wanted just to present also this, like future directions, future ideas. Yes, I know I won't be able to do all this by myself, so again, I just throw these ideas for everyone. Yeah. So again, we've observed the effect of ketogenic diet plus gemcitabine. Of course, gemcitabine is given to patients. However, currently, usually patients would receive either ketogenic gemcitabine plus Abraxane as a dual treatment, or FOLFIRINOX in many patients that are like healthier or have a, yeah. So it could be interesting to find out whether ketogenic diet alone, ketogenic diet with other treatments also enhances. Yeah, there was, as I mentioned, there was this recent paper showing the triple chemotherapy with Abraxane, gemcitabine, and cisplatin. So again, this is very interesting, that showing that other treatments or other combination treatments also enhance the survival, at least in mice. So again, this is a strict ketogenic diet. If we think of a pancreatic cancer patients, yes, with a tumor in their pancreas and maybe problems with the liver enzymes, like the compliance of a ketogenic diet could be difficult. So I think that we are starting to evaluate maybe an intermittent ketogenic diet, where we try to match like the beneficial effect of a ketogenic diet, ketogenic diet, and maybe a controlled diet. So maybe twice a week a ketogenic diet, and 3 times a week like the controlled diet, trying to mimic these ketosis levels throughout the week. And this has been shown beneficial, at least for cachexia. So if we can mimic this and maybe evaluate this, and if it improves cachexia, maybe if it improves survival, it improves the tumor biology, this could be beneficial. Yeah, other ideas is to use the ketone esters. If the ketone esters is part of the mechanism of action of a ketogenic diet, this could be beneficial. Yeah, fasting mimicking diets, this has been used. For example, Dr. Longo at USC has been working on fasting mimicking diets to evaluate this, or caloric restricted and fasting mimicking diets as a way to increase ketosis. Also, something that we are interested in is, does the ketogenic diet have an immunological effect? Is it affecting the tumor microenvironment? And if so, maybe this could also, we could provide a way to increasing or adding immunotherapy to these options. Yes, again, this is something that we are interested in, but again, I feel that it could be a nice idea to follow up to see if immunotherapy could be a benefit in this case of treatment with ketogenic diet. And also, again, our ketogenic diet was mostly lard, yeah, and the mice did not complain, but that was, yeah, our fat source, yeah, lard and some soy oil. However, what about ketogenic diets with different fat sources? Yeah, different omega-3s to omega-6. Does this affect the tumor growth? Will this have a differential effect? Yes, we are doing other studies in carcinogenesis. We are evaluating different ratios of omega-3 to omega-6. So we are seeing some differences in prevention. But what about maybe if we translate this into treatment options? I don't know. This is something that could be interesting. And of course, the most important part is moving this into patients. Yes. Again, there are some clinical trials, and again, and Dr. Cantley also mentioned that there's others as well. So this could be something interesting to pursue, trying to move this combination to see if there are some of the parameters that we've determined. This also occurs in patients. Yes, and with that, again, I will of course acknowledge the grant supports, yeah, for these studies, yeah, and this of course supports other studies as well. These are part of the members of my lab. Yeah, this, again, as I mentioned, was led by Natalia Cortez. Like now she's a postdoc. Yeah, she finished like a couple of months ago. and with the help with Dr. Rodriguez-Lancy as well, with some of the mechanistic studies. I want to, of course, acknowledge my collaborators at UC Davis. Yeah, Dr. Ramsey, that's an expert in ketogenic diets, Dr. Oteiza, Dr. Kim, Dr. Matsukuma is our pathologist, and not she's a pathologist per se. So, and then Dr. David Vasur, that he helped us with the microbiome analysis. And with that, thanks for your attention. I'll have a minute for questions, and if not, I'll leave it for the discussion panel. Thank you.
Hi, Peter Cornelius Sindevrix. You didn't show us the fat mass, did you? Or did I miss that?
The fat mass of the body composition?
Yeah.
Yes. It was increased. Yeah, it was the difference. I didn't present it here, but after this model is pretty lean. Yeah, so we saw an increase in fat mass in the body composition, but it wasn't that significant compared to the control diet. Yes, I don't know if this is the case for other models. Yes, but the KPC model tend to be a lean model. They basically did not increase, like this was given at libidum, and they do not increase their body weight. So the fat mass was not as significantly changed.
Also, I was just wondering if you measured any markers of adipose tissue function related to cancer, such as leptin and adiponectin. And finally, since I don't think Lou's here, did you measure insulin?
Yes. So we did measure leptin. There was no significant changes in leptin. Insulin, there was a decrease, and we also observed the decrease in insulin in our other models. Yes, I removed the slide of the glucose and hexokinase where I had the insulin measurement there, but we did measure insulin, and insulin was reduced in both female and male mice, and we also observed this in the KC model that we are doing for prevention as well, that the significant reduction in insulin, yes. So again, insulin, as Dr. Cantley mentioned, could play a huge role in these mechanistic studies.
Very quick question. In humans, often chemotherapy is infused in a dextrose solution. Do you know what was yours administered with dextrose or saline or what? Because obviously that would have a...
So yeah, in our case, we dissolved the gemcitabine in PBS, yes, and we injected. Yes, I know that, again, I think this is something that you usually hear, like when patients are giving the glucose or dextrose directly, like will that affect like chemotherapy and the effect of potential diets in it? And that's, maybe a problem as well. So,
Thanks.
but yeah. Okay, so we'll move on to our second speaker.